Good morning. Thank you for the introduction, and thank you to the organizers for inviting me. I'm really excited to tell you all about a story that was very recently published from my lab in eLife, the old-fashioned way, not the new way. But before I dive into that story, since I am a relatively new PI, my lab just turned 2 years old in October, I'll just give a very kind of quick overview about some of the stuff that we're doing in the lab and things that we're interested in. So broadly speaking, my lab is interested in somatosensation and how sensory circuits encode these sensations. And we use a range of techniques to address our research questions that really span ion channel biophysics all the way up to animal behavior. So we kind of have two main overarching questions in the lab at the moment. The first being how ion channels are differentially engaged in discrete somatosensory circuits, and we're particularly interested in investigating roles for understudied ion channels in the peripheral nervous system. So what I'm going to talk to you about today is going to be about voltage-gated sodium channels and proprioception, but we're also interested in voltage-gated sodium channels and thermosensation and pain, as well as how large pore channels like connexins and pannexins that release ATP can contribute to pain signaling and neuroinflammation. And then in a different line of investigation, something a little bit more in the long term that we're really interested in tackling is how peripheral nervous system signaling impacts and interfaces with brain function. So we're really curious about this peripheral central crosstalk. And so we'd like to know, for example, to what extent do sensory neurons control brain development and behavior? And as you'll see a little bit later in my talk, we're also very interested in understanding how human sodium channelopathies and sensory neurons can help contribute to the clinical manifestations that are typically associated with brain disorders. And then briefly, as Raj mentioned, I do write kids' books. I'm the author of The Magnificent Makers and the Ada Twist Scientists, the Y-Files series. I'm just really passionate about diversifying science, and I really think that begins in early childhood. Starting in grad school is way too late. And so that's a big motivating factor for why I do this kind of side work. But back to science for adults. So I will again tell you, I will be talking today about how voltage-gated sodium channels contribute to proprioceptive sensory transmission. That was kind of one of the questions that my lab set out to investigate. So first, I just kind of like to talk about what exactly is proprioception. So fundamentally, proprioception is our internal spatial awareness of self. It allows us to distinguish ourself from somebody else physically. So I don't know if anyone else here has a parent of maybe a small child, but I have a 4-year-old daughter, and I would say about 95% of the mornings that I wake up, I have a limb across my body that is not mine. And the reason that I know that it's not mine and is hers is because of our proprioceptive sensory network that, again, allows us to internally distinguish ourselves from somebody else. And this internal sense of self is really required for purposeful movement. And so it's required for these kind of complex movements that you see here on the screen with Simone Biles and Tiger Woods, but also for just pretty much anything, walking, sitting up straight at a desk and holding your posture. All of this requires normal proprioceptive coding. But unfortunately, we actually don't know a ton about how these signals are sent and transmitted by peripheral sensory neurons. So about, I forget, maybe 5-7 years ago, time flies really fast, Ardem Pataputian discovered that piezo2 mediates proprioceptor mechanotransduction. So I'll just briefly show, I'll just briefly say that these proprioceptive signals are carried by these dorsal root ganglion neurons. And so many of these neurons will innervate peripheral targets such as the skin. However, proprioceptors innervate skeletal muscle, and they respond to changes in muscle length and muscle force. And indeed, it is the ion channel, the mechanosensitive ion channel piezo2 that are detecting these movements. And when you delete piezo2, either specifically in parvalbulum expressing neurons like proprioceptors or in lumbar spinal levels, you get these very abnormal limb positioning, very awkward gait, very clear proprioceptive deficits, and you also, at the afferent level, have the inability to transduce muscle movement into an electrical signal. But to date, this is kind of the only channel that's really been, in my opinion, convincingly shown in vivo to be required for proprioceptive transmission. And so, based on some of the postdoctoral work that I had done on voltage-gated sodium channels, I became particularly interested in how voltage-gated sodium channels, which as we know are required for action potential generation and propagation, which are the isoforms that are contributing to proprioceptive signaling. And so, for this audience, we don't need to go over this in too much detail, but just suffice it to say that there are 9 mammalian voltage-gated sodium channel isoforms, and in the healthy adult peripheral nervous system, 5 of those are expressed: NAV 1.1, 6, 7, 8, and 9. And although there is some subtype specificity, broadly speaking, as you can see, they're kind of broadly expressed, at least at the mRNA level, across sensory neuron populations. Now, voltage-gated sodium channels contribute, again, as I said, to action potential initiation and propagation, and we know a little bit about how the different isoforms do this. I know Raj mentioned in his talk that NAV 1.7 contributes both to subthreshold stimuli as well as the rising phase of the action potential. You have 1.6 and 1.8 that are primarily contributing to that rising phase, and then 1.9 is a subthreshold booster. 1.3 in injury also works as a subthreshold booster and is upregulated even though it's not normally expressed. But one thing that you'll notice is that there's an isoform missing from this one, and that is NAV 1.1. And that's largely because NAV 1.1 is pretty understudied in the peripheral nervous system. Now, it has widespread expression in the central nervous system, including places like the cerebellum, where it contributes to motor behaviors. And the gene that encodes NAV 1.1, SCN1A, is actually referred to as a super culprit gene. There are about, I think the most up-to-date number is roughly 3,000 disease-causing mutations that are associated with this locus, the majority of which produce a profound developmental disorder called Dravet syndrome, which is accompanied by severe epilepsy, intellectual disability, motor dysfunction. You know, it's a very severe disease. And so it's likely that this prominent role that NAV 1.1 is known to play in brain function and brain diseases has likely taken attention away from its role in the peripheral nervous system. Nevertheless, it is found in discrete sensory neuron subtypes, including cold receptors, which is actually what I found as a postdoc, mechanical nociceptors, and proprioceptors. So this is a muscle spindle. It's an end organ that is formed in skeletal muscle by proprioceptive terminals. And this is staining against NAV 1.1, and it was found to be really kind of highly expressed in these endings that wrap around muscle fibers to detect muscle movement. But this was the only paper when we embarked on this project that had localized sodium channels, protein at least, to these neurons. There's some RNA sequencing data sets out there that shows it's expressed, but no one had looked at this functionally. So the first thing we did is a post-bac researcher in the lab, Cheyenne Lewis. She did some RNA scope on a reporter mouse line, a parvalbumin TD tomato reporter, which in the peripheral nervous system will label proprioceptors. We probed for NAV 1.1 and actually found 100% of neurons were positive for NAV 1.1 transcript. So, of proprioceptive neurons were positive for NAV 1.1 transcript. So that was a good start. But we also know that other sodium channels were there, and so we wanted to know what was the relative expression levels between these three. So in addition to 1.1, we also knew that 1.6 and 1.7 were also found in proprioceptors. And interestingly, we did find that 100% of those proprioceptors also expressed to some degree 1.6 and 1.7. However, when we looked at the intensity of that signal, we found that NAV 1.1 was significantly higher expressed, at least at the mRNA level. So this is work that was done by an undergraduate in the lab, SNIGDA. But my graduate student, Cyrus, he wanted to know, okay, this is mRNA, what's going on actually at the functional surface protein level? So here we turn to patch clamp electrophysiology combined with some selective or decently selective pharmacology. And so we recorded from these genetically identifiable proprioceptors, and then we did an experiment where we serially blocked the different channel subtypes. So we used an ICA compound to first block NAV1.1, then we followed with this AHTTX compound, which is reported to be selective for 1.6, but actually also hits some 1.1. But we kind of reasoned that since we've already blocked 1.1, we can use it as a more selective 1.6 inhibitor, and then a blocker of NAV 1.7. And consistent with our RNA scope data, we found that the ICA sensitive component, which likely represents NAV 1.1 channels, was the dominant isoform, at least from these somal recordings in vitro. Next, we had NAV 1.7, which was roughly a third of the whole cell sodium current, and 1.6 only seemed to make up about 15% of the whole cell sodium current. But it's good to note that we did have around 8% of the current on average that we couldn't block, highlighting the fact that this is probably, a little bit of dirty pharmacology. Okay, so the next thing we wanted to do is just a general characterization of the whole cell sodium current in proprioceptors, because this really just hadn't been done before. No one had looked at sodium currents in these neurons. And so again, my grad student Cyrus just did a basic analysis. We analyzed the current voltage relationship, where we found that sodium currents in these neurons start to emerge around -50, peak around -30. We performed activation and inactivation curves and saw the V-half max of activation was around -40, for inactivation around -65. Interestingly, these neurons recovered really quickly from fast inactivation with a tau of just over half a millisecond. So we were actually a little bit surprised it was so fast. And then we also quantified entry into slow inactivation, where we found a tau of a little bit over 900 milliseconds. So somewhat refractory to entry into slow inactivation. But then we wanted to know kind of what NAV 1.1 was doing here, and so we kind of repeated some of these voltage clamp experiments where we again saw this more or less 50% reduction in the whole cell sodium current that was accompanied by a slowing of the whole cell sodium current's decay kinetics, consistent with inhibition of a channel with very fast gating kinetics like 1.1. In current clamp experiments, we saw a very significant reduction in action potential firing. We typically would only have 1 or 2 residual spikes at the beginning of the stimulus, and we really completely lost the ability of the neuron to repetitively fire. And this was accompanied by a reduction in action potential amplitude, a hyperpolarization of the action potential threshold, and a significant widening of the action potential, so an increase in action potential duration. Again, all consistent with lost inhibition of NAV 1.1. So this is all fine and dandy, but we really wanted to know, in vivo, behaviorally, is NAV 1.1 relevant to proprioceptive signaling? So we created a mouse in which NAV 1.1 was deleted in all sensory neurons. We're not able to specifically target proprioceptors because the Cree driver used to do that will also hit brain neurons that express NAV 1.1 and basically creates an epileptic mouse. So we instead took the approach where we just deleted it in all sensory neurons. And so this is what, for anyone who might not have worked with mice before, this is kind of what a typical mouse does when you pick it up by its tail. But conversely, you can see our knockout animals have very notable motor deficits, these kind of shaky, ataxic-like movements. They also have very abnormal limb positions, which you'll see right here in a second. Right there, you see the mouse just completely turns its paw around. And then in this final video where I'm scruffing the mouse, again, you can see its paws are just completely backwards. It's almost as if it's trying to feel its way out of my grasp, as opposed to being able to internally sense its body to try and escape. So this was exciting because it suggests that indeed NAV 1.1 is contributing to proprioception or motor behaviors in vivo. And we quantified this first using just the open field to look at exploratory behavior. And interestingly, we found that the conditional knockout animals moved more slowly, as well as moved less, despite not spending a different percentage of their time moving compared to the controls. But what was really surprising, and actually quite exciting for me, was that the heterozygous animals were actually identical to the conditional knockouts. So they also moved more slowly and moved less compared to the flock's controls. Now, when we looked at the rotarod, the conditional knockout animals performed horribly. They fell off about 50% faster than the flock's controls. But in this scenario, the het animals performed like the flock's controls. So they were able to do this task without any major difficulty. So what this suggests is that the het animals are very much het. And this is important for one reason. So as I mentioned earlier in my talk, NAV1.1 is a superculprit gene, has all of these mutations that lead to Dravet syndrome. And one aspect of Dravet syndrome is ataxia and motor developmental delays. And all of these patients are hemizygous for the mutation. So they have just one loss, one copy is lost in these patients. And so what this is suggesting is that at least some of those motor deficits are not just solely due to abnormal brain function, which has been reported predominantly in the literature, but this provides evidence that aberrant sensory neuron signaling, specifically aberrant proprioceptive signaling, is indeed contributing to some of the clinical manifestations of this rare childhood disease. So that was quite interesting for us. We also looked early in development. So these are pups that are about P7 or P8. And again, you can see your flock's control kind of doing what it normally does. However, if you look at the conditional knockout animals, you can just see some just very, kind of odd movements. And you can see a little bit of that shaky behavior here as well. So it would appear that NAV1.1 is required very early in development for the normal development of motor function. So in just the last few slides, I'll say we looked anatomically to see if deletion of NAV1.1 was interfering at all with proprioceptor development. We didn't see any qualitative changes in muscle spindle formation, which is consistent because when you knock out piezo2 and also get these motor deficits, they also didn't report any changes in muscle spindle structure. We used RNA-scope to quantify the number of proprioceptors in DRG, so we probed for parvalbumin and RUNX3, and colocalization of those two markers is kind of a bona fide a signature of a fully developed proprioceptor, and we didn't see any difference in the number of proprioceptors across genotypes, so we don't think that loss of proprioceptor development is contributing to this. We also looked at proprioceptor innervation of motor neurons in the spinal cord, and also didn't see any significant changes. So we don't think there's any or at least the way we did it here, no super obvious circuit rewiring, although I'll say at the end, we are interested in looking at that functionally. So the last thing we did was ask, okay, the electrophysiology that I showed you earlier were somal recordings that were done in vitro. But when we think about proprioceptive signaling, the signal is being sent down axons. And it's not super clear, what the soma in DRG is doing to that signal. So I collaborated with a woman, Dr. Catherine Wilkinson at San Jose State, who does these really beautiful and elegant muscle nerve recordings. So you just isolate out the muscle and do these extracellular ex vivo recordings from the nerves that innervate it. And she's able to get these beautiful single units. So this is what a typical response would look like. You stretch the muscle, you get this kind of initial burst, and then a sustained firing throughout the duration of stretch. And in our floxed animals, we found that the vast majority of them, consistent with what's known in the literature, will fire the whole time during stretch. On the other hand, if we look both at heterozygous and conditional knockout animals, what we found was inconsistent and unreliable firing. So it might kind of start in the beginning, then you get these failures here, or as you can see a very noticeable case here, you see an initial stretch, then it stops, it bursts again, and then just stops. And so we didn't notice like a really regular pattern. It wasn't like the afferents just fired always in the beginning and then stopped here. But what we did see was this very unreliable and inconsistent firing. And what we think is going on in vivo is that the muscle, without NAV1.1, the muscle is, the proprioceptor is unable to maintain that repetitive firing, kind of similar to what we saw in vitro. On the other hand, when we look at dynamic stimuli, so response to vibration, we saw no genotype dependent differences. So even up to 100 hertz, NAV1.1 knockout animals were able to fire action potentials that entrained to that sinusoidal wave. So it's not like there's a complete loss of electrical activity or excitability. It does seem to be a specific deficit in muscle stretch. And so that kind of brings us to the last slide, which is the current model that we're working with. So what we think is going on is that in response to muscle movement, you have opening of piezo2 channels. This is going to activate NAV1.1, and I have this as 2 and 3, but more or less all at the same time. So NAV1.1, 1.6, and 1.7. What we think NAV1.1 is doing is boosting those receptor potentials that are generated out in the periphery. And that is going to allow for propagation of proprioceptive signals faithfully and reliably into the spinal cord. And in the absence of NAV1.1, we see this inconsistent and unreliable firing that results in abnormal motor behaviors. We're really interested in figuring out what 1.6 and 1.7 are doing. One idea, we know in the brain, NAV1.6 is localized often to nodes of Ranvier, so we're very curious as to whether or not 1.6 might be playing a more direct role in transmission, and we're actively investigating that. And we're also curious as to whether or not aberrant proprioceptor firing reduces neurotransmitter release from presynaptic terminals in the spinal cord. While we didn't have any evidence for anatomical changes, we're very interested in whether or not there could be potentially functional changes that result from loss of NAV1.1 and proprioceptors. So I basically summed that up all here, so I won't go over that again. And again, this is just the video of that poor mouse. I do want to make two very, very quick plugs if I can. One is for all, since I'm an ion channel crowd, I thought that I would just make you all aware of this wonderful initiative that I launched in collaboration with the Biophysical Society. It's called Justice for Underrepresented Scholars Training in Biophysics, or Just B. We had our inaugural poster session at the 2022 Biophysics meeting. It was a huge success. So if you are going to be presenting a poster at Biophysics and are part of an underrepresented group broadly defined in biophysics, please do apply and register to present in the Just Be poster session. And then lastly, I am organizing a conference for the Society of General Physiologists on pain in 2023 at Woods Hole, and we actually have 2 speakers for this meeting here in the audience. So if you're interested in ion channels and pain, please do keep an eye out for this. And with that, I'll take questions.
Theanne, beautiful talk. Congratulations. I may have missed that part, but so NAV1.1, is it expressed in other DRGs, other DRG neurons, other than proprioceptive? And the question is, because you use the PERT mouse you took out from everywhere. So the question is, do the mice have other deficits? Light touch, pain, whatever.
That's an excellent question. So yes, NAV1.1, I didn't show it here, but we quantified it across populations. We saw about only 3% of non-peptidergic nociceptors expressed NAV1.1 mRNA, about 24% of peptidergic nociceptors expressed NAV1.1 mRNA, but 93% of myelinated neurons, if you just kind of group them all together, so your touch receptors, expressed NAV1.1. So it's very possible that our mice could have some tactile deficits that are contributing to the kind of overall phenotype. However, I will point to one thing in this video really quickly. So if you look at the mouse, as soon as it grabs on to the bar, it kind of stabilizes, and that kind of shakiness goes away. And so I wonder if touch actually isn't that impaired and actually might be kind of stabilizing the lack of internal sensing by this kind of external sensor. And that's also consistent with pharmacological data where you inject a NAV1.1 antagonist into the hind paw. It did not change von Frey mechanical withdrawal thresholds. So it might be, but we are definitely exploring approaches that allow us to specifically target proprioceptors using intersectional strategies. I worry about doing a Von Frey in this mouse, though. Like, I'd be a little nervous about those withdrawal latencies.
So really neat. I was fascinated by the differences in the hind limb responses and the forelimb responses, especially in the pups, where some of them would be crossing the hind limbs and others wouldn't. There's some modification of information crossing the spinal cord to coordinate those?
Yes, I mean, that's exactly the kind of questions that we're hoping to ask by getting into spinal cord electrophysiology. I don't have a great answer for you at the moment, besides to just note that in the piezo-2 animals, you also tend to see this hind limb phenotype more than you see it in the forelimbs. And so I don't know if that says something about the proprioceptors that innervate the forelimbs as opposed to those innervating the hind limbs, or those that are at thoracic levels versus lumbar levels. But these are the questions that we're very curious. There's 3 known functional groups of proprioceptors, but anywhere between 5 and 8 molecular classes that have been identified using single cell RNA sequencing. So we're trying to kind of really tease that apart.
And do you know if 1.6 and 1.7 also contribute to proprioception?
My goal for my lab over the next couple years is to turn all of these kind of dashed arrows into nice solid arrows, to kind of like remove the question marks, or just maybe take away the arrows if they're not actually involved. But I'm curious. It's my understanding that the 1.7 knockout animals don't have like an overt motor phenotype. So I don't know what 1.7 is doing there, but we found it everywhere. And at least at the soma, it seems to, be a notable component, but I'm not sure.
Yeah, I have. That was great. Thank you, Theanne. I have a really quick question, and it's pretty predictable for me. But have you looked at the different splice isoforms and to see? I mean, it's quite likely, right, that you've got some pretty beautiful cell specificity in terms of expression patterns, and so it'd be really great. I just looked quickly, and there's a lot of really nice splicing going on in there.
Absolutely. We have not dove really hard into those kind of genetics just yet. But again, these are all the things that we're very excited to look at in the future, especially for NAV 1.1, given its notable disease role. Thank you.
That's a R01 idea right there. Thank you very much.
Thank you.